Okay, so our next talk today is Dr. Penny Ajani, who will be presenting the second stage of the Oyster projects that Shauna spoke about earlier this morning, but this one is called Transforming Australian Shellfish Production. Now, Penny is also at the UTS, and this project, as Shauna alluded to earlier, it aims to transform the shellfish safety and disease response in two major production regions, New South Wales and WA. So this will directly deliver around $800,000 per year in improved regulatory practices to enable savings of around $5 million per year in reduced losses. So Penny, I'll hand it over to you, but I see Shauna, you're on board as well, just in case we do need to throw to you as well.
Great, thanks Nick, and thanks David and the Food Agility for inviting us today to present our work. Shauna has already introduced, as you say, the stage one of our project, and based on the success of that project, I'm now going to talk about stage two, which we've called Transforming Australian Shellfish Production. As Shauna mentioned, this is a collaborative work, a project. We couldn't be doing this without the oyster farmers themselves, but we also have the Department of Primary Industries from New South Wales, the Food Agility, researchers at the UTS, and going into stage two, we are now very excited to have the New South Wales Farmers Association on board and the Western Australian Department of Primary Industries and Regional Development. So as we mentioned, it is a huge collaborative project, but what we really aim to do is to use state-of-the-art research and sensing to deliver real-time water quality testing to the oyster farmers. And we hope that this will improve the efficiency and transparency of their regulation, and hopefully also to provide better informed and more responsive management. As Shauna mentioned, shellfish farmers in Australia are not currently able to predict harmful risk or high risk events. And if the farmers were able to do that, they could take meaningful action to either harvest early or to shift their stock to lower risk areas. We, in this stage two, which goes from 2001 to 2024 and is a $1.5 million investment project, we aim to use the backlog of all the samples that were collected during phase one and to deliver some tailored models. And we will be doing this on an estuary by estuary basis, as all of the estuaries in New South Wales have different issues and different problems. And today I will present a few case studies from some of those estuaries. The stage two will also bring on board two new estuaries, the Macleay River to the north and Merimbula to the south. Very exciting, we are also bringing on board Western Australia. We are delivering 4 sensors into Oyster Harbour, Albany, and we're very excited to have them on board. We also hope to embed these models into existing oyster management apps, and I'll talk a little bit about those further along. And finally, we are setting up a data portal so that the data that we have collected will have more broader use, both for water quality issues and public health. So the first case study that I really want to talk about was a harmful algal bloom that occurred in Wodonga Inlet in October 2019. And this estuary has experienced these harmful algal blooms before. They've had major closures due to these blooms, and they've lost millions of dollars. So we took this event as an opportunity to test some hypotheses in relation to the sensor data that we're using and collecting, and also with our novel molecular assays that we're developing. So we went out, we collected samples during this event, We used electron microscopy and DNA sequencing to actually identify the culprit. And we know that this species is a toxic diatom belonging to the group called Pseudonychia. This group produces domoic acid, which is a neurotoxin, and places like California have some really major issues with this particular group of diatoms, where they can have hundreds and hundreds of marine mammals washed up on the beach, either poisoned or dying. So we then developed a qPCR assay to measure the DNA from this particular species. We then took the abundance of that data, of that particular phytoplankton. We combined it with the high frequency sensor data that you can see here that's collected. There's a picture of the actual sensor. And we combined that with rainfall data, and we did some modeling. And what we found was that the high frequency data could predict that harmful abundance way better than the rainfall data that's previously been used. The second case study is set in the Manning River, which is to the north of the mid-north coast of New South Wales. And what we did here is that we tracked E. coli over the last 2 years. This was based on the water samples that the oyster farmers collected. And we developed a rapid qPCR assay to detect this E. coli. And that you can see with the green line going along here. And the yellow bars are rainfall events. So we actually asked the farmers to collect some high frequency data, samples when these rainfall events occur. We then also successfully developed these assays to detect the various types of E. coli data that could be coming into the river system. So we developed one for birds, cattle and human bacteria. And you can see there the same time period, 2 years worth of data. We show the bird E. coli, the bird bacteria data across that time period, the cow and the human. And what we can see from this is that the bird bacteria is quite random. It doesn't really follow the rainfall events. The cow does follow the rainfall events. And the bacteria, and the human is slightly random, I guess, and fairly low, which is a good result. As I said, we asked the farmers to collect some high frequency rainfall event samples, and you can see that in the plot on the right-hand side. That's 4 different rainfall events. And during the course of our project, the oyster farmers actually collected 10 rainfall events. So the more data we can get and the more we can model this data, the more likelihood we can predict and show the patterns that can occur during rainfall. What we can see from those 4 plots on the right-hand side is that the bird and the E. coli, the cow and the E. coli pretty much drop off after 3 days of rainfall, so that's good. The bird is variable, and we think that the bird bacteria entering the waterway may be more related to things like migration and breeding of the birds, and possibly even the feeding of hot chips to the birds by the tourists at various times during the year. But as I said, the more rainfall events we can get data on, the more we can predict these kinds of things. The models on this data has shown again the proof of concept that the sensor data is a far better predictor than the rainfall data, which is currently used for management practices. Also, the modelling data has shown in this estuary that high salinity equals far better oyster growth. And that may be something that the oyster farmers already know, but it's, or, you know, have maybe know from years of practice, but it's really good for us now to have that kind of data, and they can take appropriate management of their harvest accordingly. The last case study I want to talk about is the impact of the black summer bushfires on trace metals in oysters. These fires burnt out approximately 7% of the whole of New South Wales. That's about 55 million hectares were burnt. 37% of all national parks were burnt, and 42% of New South Wales state forests were burnt. And this was mainly along the eastern seaboard of Australia, as you can see by this gruesome map. And while a lot of work has gone into the statistics behind those numbers that I just provided, there's been very little work on the impact that those bushfires had on the coastal ecosystem. And we are in a unique position with our project in that we have both before, during and after samples. So what we aim to do is, as Shauna mentioned, we deployed oysters at the site of the sensor in all of the estuaries, and we retrieved 3 of those oysters on a weekly basis and froze them. We are now going to examine those oysters for both from both impact and control locations. We've selected 3 of each, and we're going to look at them for a suite of trace metals, which is something that is in the literature that can be an effect from bushfires. So stay tuned for those results. We're you know, in one way excited to get these sort of preliminary bushfire results on our waterways, because there's just very little information around, and you know, with all climate change models, we expect that this will be these catastrophic events will occur into the future. I also mentioned that we have 2 new estuaries on board. So for the second stage of the project, the Macleay River in the north is a river that has a high residence time. So it's affected by both stock and sewerage contaminants. We are also looking to work with Merimbula. Well, we are working with Merimbula. We've deployed sensors in both of those. Merimbula is adjacent to another estuary, Pambula, and they looked across to Pambula and saw the success that we had in stage 1 of the project. And so Merimbula saw what their neighbours were doing and wanted to come on board for stage 2. And finally, the 4 sensors that we've just put into Albany in Western Australia. And I don't think this video is going, oh yes, it's going to work. I'll just fast forward it. But this was the deployment of the 4 sensors into Albany a couple of weeks ago. Unfortunately, we couldn't be there due to COVID restrictions. But we heard from Deborah Gardner, our WA collaborator, that it was a beautiful sunny day and all 4 sensors were deployed without a hitch. So we're very excited about that. And that's our little, one of our 4 minions that's just gone into Oyster Harbour, where I might say they harvest both oysters and mussels. And that's the data, real-time data that's coming through from those sensors. We start our biological sampling in a couple of weeks, and that will go for the next 2 years before we can begin to model that particular estuary as well. We aim to, once we've developed these models and where we get, as I said, all the estuaries along the coast are vastly different, but where we do get some successful and high predictive models, we hope to embed these into existing Oyster management apps. We have now engaged with this company called Oyster Cloud, and this is just a screenshot from their app, which is a cloud-based web application, and oyster farmers use it to manage their stock. And what we hope to do is to have a very simple system like a red light, green light, amber light sort of warning system for a high risk time or possibly a closure. So in the first stage, the Oyster app company will just have the real-time data available, and they're working towards that at the moment. The second stage they will have alerts to have closures in response to rainfall events. And the third stage of this, we hope, is to develop our models, our biological models, and that will provide some warning for something like a harmful algal bloom. And finally, we want to make this data available we've already had requests from various universities, from various local and state governments. And so we are now working with the UTS research team to package this data up and have it available to consumers or users. They'll have a login. They can extract the data in any which way they want, whether it be by estuary, by year, by variable. So we're hoping to get that up and going fairly soon so that we can distribute that data as freely as is requested for both public health reasons and for research reasons. So just to summarise, the Transforming Australian Shellfish program aims to deliver harvest area closure notifications embedded in these existing management apps. We aim to provide predictive information on contaminants and algal blooms in real time, and we aim to provide insights into the optimal conditions for oyster growth as we learn from these models along the way. And we hope that ends in greater yields for the farmers. And as well as thanking all of these beautiful faces below, as Shauna mentioned, we couldn't do this work without the oyster farmers themselves. So thank you to them, even though they're probably not listening. Thank you.
Thank you for that, Penny. It's really good to see where this new project is going and how it is being scaled into WA as well, and hopefully getting some really good insights from over there. I've got I guess one question that is around the bushfires, and you said you're in a unique position to look at the impact of bushfires over the industry, particularly in New South Wales. Is there scope to essentially look back at when you do sort of look at that data, scope to look back and somewhat assess the severity of bushfires as to what their effect would be? If it was a small bushfire, what your potential impact on the industry is going to be, as opposed to what a large bushfire's impact would be on an adjacent estuary?
Yeah, I'm sure we have the capability of doing that. I think it's going to depend on the catchment that was, the size of the catchment that was burnt, but we definitely have that sort of information, and we could do it on an estuary by estuary basis. We have an incredible backlog of samples now in the freezer in terms of, we have 8,000 samples. And as I said, we are in that unique position where we have before, during and after. No other project I know has that kind of time series through those bushfires. So we can have a look at exactly, individual catchments and see what was going on beforehand and what happened after. So yeah, the world's our oyster as far as those things are concerned, so to speak.
Yeah, absolutely. And I think, that could be some really, really remarkable results come from that, hopefully, and just see what it eventuates. I guess for this project so far, it is a new one. So, what's your eureka moment so far within this project?
Yeah, I was talking to Shauna about that before, and I mean, unfortunately, I feel like the gnarly and the eureka came at once with those bushfires, in that we realised, first of all, how devastating it was for the farmers. Shauna mentioned that some of those farmers were actually fighting fires at the same time as trying to save their oyster leases and their homes and all of that. So that was gnarly, but the eureka that came out of that was that we do have these samples now, and I think they will be gold into the future as far as we can do with them, and what we can inform the oyster farmers about the impact of contaminant. I mean, there's just very few studies anywhere in the world that shows the impact of bushfires on coastal waterways. And a lot of people rushed out after the bushfires to take samples, but without the before and during, it's not a complete story. So
Yeah, and I guess, as you say, because you've got those before, during, and after samples, you're able to actually track the level of those heavy metals that you might be able to pick up within the oysters, and whether certain ones are going up and certain ones are going down, or the levels that they change. So it's going to be a really interesting data set, as you say, going forward, and some key research to come out of that.
Yeah, just Mick, I'd say another thing too, that that was not really our UTS kind of eureka moment, but I think it's really helped for the DPI aquaculture research, has been the impact on their research on oyster selective breeding. So that was more of a byproduct, because we just wanted to deploy oysters across all of our estuaries, which were genetically identical, so that we'd know that any difference in growth was not related to genetics. It was just related to conditions happening in the estuary at the time. And so they gave us their kind of top of the line selectively bred oysters for that, and deployed it all the way across. And now they've been able to have those sort of 2 years of data on these top of the range selectively bred oysters and how they perform in the various different estuaries. And then I mentioned in my talk that there's been a QX disease outbreak in the last few months. And so now they're able to take those data and show the industry, look, we've actually got data on how our selectively bred QX resistant oysters perform in your estuary. And we've just done it over the last 2 years. So that's really been a byproduct, really, but it's really worked out. It's really valuable for them now.
One other thing I think is really interesting is that we may be able to, just because we have a statewide data set, we may be able to come up with some more kind of groupings that, depending on the type of estuary that we've come across. So do oysters grow better in a particular type, a smaller estuary, a faster growing, a faster flowing estuary? Just once we get that whole statewide picture, I think there'll be some really interesting conclusions out of that.
Yeah, absolutely. And I guess moving over to WA as well, it's a different estuaries again over there, and just being able to compare the two different sides of Australia, really, as to how the oysters grow and how they perform in potential contaminants that you might be picking up in different estuaries in different areas. So there's a lot to be gained and learned from this sort of research. So it's something obviously very valuable to the whole industry.
Absolutely. We're going to take on the rest of Western Australia as well.
Yeah, well, it's a brand new industry in WA. So I guess they're kind of, they don't have a kind of system already in place like we had in New South Wales. They're really starting from scratch. So it's kind of exciting in a way, like they're starting straight out with the sensor, and I think that we'll learn a lot from that.
Yeah, absolutely. It's really good that they're able to start with the sensor, as you say, and build on knowledge that's been gained in the last 3 years from the past project, and then obviously the start of this one. So it is looking up. Look, thank you very much, Penny and Shauna, for those insights to this project. And we're obviously looking forward to tracking this as it develops in the next couple of years. And yeah, thank you for your time and presenting here today to the symposium.
Thank you. Thanks for having us.
Thanks, Nick.
Thank you.